This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 34, for May 22nd, 2009. From New York City, I'm Vincent Racaniello.
And from Western Massachusetts, I'm Alan Dove.
And our guest today is Stephen Morse. Welcome to TWiV, Steve.
Hi, thank you. I'm delighted to be here.
We've been meaning to get you on the show for so long because, as you know, one of the hosts of TWiV is your old friend Dick DePommier.
Yes, we're partners in crime. We teach a course together in emerging infectious diseases in the graduate school. And that's been a lot of fun, actually. I have to say, This Week in Virology never runs out of material. There's always something happening.
You know, I can start the night before to prepare an agenda, and I have plenty of material. There's never a paucity. And of course, these days, there's even more. And Steve, before we go on, Steve is a professor of clinical epidemiology and the founding director and senior research scientist at the Center for Public Health Preparedness, the Mailman School of Public Health, Columbia University, which is just across the street from us here at the medical center. And you are one of the original founders of ProMed Mail, one of our favorite sites.
Oh, well, thank you. I'm glad you like it. We actually it started in kind of an unusual way. We had a lot of people on our ProMed steering committee, the purpose of which was really to try to bring people together from all over the world to develop a better system of global surveillance, early warning, and response for emerging infectious diseases. And it's actually a funny story because way back then in the early 1990s, everyone in the world thought that was WHO's responsibility, except, of course, the people in Geneva who didn't think it was part of their job. So we decided to get a group of people together public health people, scientists working in laboratories and so on, to figure out how we could actually develop a network for surveillance to improve capabilities worldwide. And we gradually came to realize, not so gradually, I very quickly came to realize that every time I tried to convene a meeting, everybody had some different means of communication. So perhaps out of frustration, we decided to get them all on email. And once we started talking on email, we then decided to make it a listserv open to everyone. Since then, it's grown. I think it has about 55,000 subscribers in most countries of the world, and always glad to have more. It's free.
How long ago was this that you put together?
It started in 1994, although we had the email among the group a little bit earlier. But I have to admit, it was a steep learning curve at that time to do email. It's improved quite a lot now.
But now I access it on the web, which is the way I presume most people do.
Oh, yes, and it's that's been a very handy thing. So you don't have to keep all the old emails if you want to track a thread.
But what's on the web is all the emails,
everything.
We don't miss anything by not getting on the list, right?
No, everything's archived. There's also a sub-list called EDR for Emerging Disease Reports. If you just want sort of the latest news, the sort of headline items rather than discussions on all these odd infections, which are always interesting.
No, I think they're great, especially with H1N1 lately. There have been a lot of interesting discussions, and I think it's fabulous. It's a great resource. So we're indebted to you for doing that. Now, I'm just curious, you're involved in pandemic preparedness, disease outbreak response, but at one time, were you a virologist?
Yes, I actually was. I was originally trained as a virologist at the University of Wisconsin.
With whom?
R.P. Hansen. He actually worked on some paramyxoviruses, including Newcastle disease virus. He worked on our group also with some authentic orthomyxoviruses and with the spongiform encephalopathy agents. Needless to say, I chose the more difficult path and thought the spongiform encephalopathy agents were really interesting. I really recommend this to anyone who has a suicide wish, if you have a death wish, because they're very slow experiments. They take 6 months to do, but it's really fascinating. And as you know, some years ago, Prusner determined after I was safely out of graduate school that this appears to be a misfolded protein which he calls the prion protein, that's responsible for diseases, spongiform encephalopathies like Scrapie and Creutzfeldt-Jakob, and of course mad cow. So it's been an interesting field and continues to be. But I've done more conventional viral immunology. I was at Rockefeller before I came here.
Which is where I met you, I think.
Very likely.
I'm curious, and probably listeners are too, how you made the transition from research to policy.
Yeah, and it was actually a very hard decision because, once you get out of the lab, obviously, it's a completely different life. I got very interested in this whole question of emerging viruses. Actually, I blame Josh Lederberg for that, or give the late Josh Lederberg, who was then president of Rockefeller University, credit for asking, in his inimitable and typical fashion, a couple of questions that just, led to my thinking about this whole question of, where do these unknown and unexpected infections come from and why do they happen? And I became more and more interested in that. The late John LaMontagne at NIH and I co-organized a meeting that NIH sponsored in 1989. It was on May 1st, 1989, so we just passed the 20th anniversary of the first meeting ever, I guess, on emerging viruses. And what we discovered there was that we knew many of the reasons. We just, had never really brought all the different pieces of knowledge together. So some people looking at evolution thought it had a lot to do with, the evolutionary potential of these viruses. Others saw zoonotic transfers. And in many cases, we know many of these emerging viruses now already exist in nature. They may continue to evolve. One of my old professors, Howard Temin, who won the Nobel Prize for his work on reverse transcriptase and retroviruses, and sadly died much too young at age 57 a few years ago. Howard used to say that when I would highlight influenza as an example, he used to say, that's cheating, It's not an emerging virus. And I said, well, why not? It keeps doing it. And it exemplifies everything that we talk about, the element of surprise, the unpredictability, the origin in other animal species, and the continuing evolution. So if there's one virus that probably exemplifies everything we're talking about in one package, influenza, mundane as it may seem sometimes, is a very good example.
So did you coin the phrase emerging virus?
I thought I did anyway, so and I defined it, so I take the blame for it.
Very good. I thought you had. And I asked Dick, and he didn't know. He said I should ask you. So straight from the horse's mouth. Alan, are you still with us?
Yes, I am. I'm right here listening. It's fascinating stuff.
I just want to make sure we haven't lost you, because modern technology isn't perfect. Well, in fact, influenza is the story of the past month, and we have quite a few stories about flu today. So let's get right on to our stories. The first is actually not influenza. We have one non-influenza story. We try and keep a perspective here on TWiV. And this is, unfortunately, a sad story about progressive vaccinia in a military smallpox vaccinee. And this is a case where a young man in the military, 20 years old, in January of this year, received the Acambus smallpox vaccine, which is is it a live attenuated?
Yes.
It's a vaccinia-based vaccine, right?
Yes, and of course, as you know, in this country, we use a strain that was originally derived from the New York City Board of Health many years ago. In some other countries, they're using one called MVA, which is considered gentler and milder. It doesn't actually make as many rounds, may only make one round of replication. And the question being, what's safe enough to immunize immunosuppressed people like HIV-positive patients?
Sure. So this gentleman started having symptoms of being ill. 12 days afterwards, he had fever and headache, and the rash where he had been immunized was growing larger. And it turned out that he had acute myelogenous leukemia, which they found out after he received the immunization. So he should have never received the immunization at all because he's immunocompromised. And they had to give him chemotherapy, 2 rounds of the chemotherapy, and then they gave him immune globulin, a huge number of treatments. The number is escaping me right now, but it was enough to treat many, many people, apparently, what he received. And eventually he became well, but unfortunately they had to amputate his 2 feet because of various problems on vasopressor support, intubation. So this is really a tragedy, and it goes to show that you shouldn't give a live vaccine to immunocompromised individuals. And I think we should point out that this is a vaccine which is currently just routinely given to the military, right?
Right now, yes.
Because they may be under threat from bioterrorist attack of some sort, I suppose, right?
Yeah, it's kind of ironic, as you know, smallpox, which was a great scourge of the past. Some people have said that it's killed more people than all the wars in history put together, and it's certainly been with us for, at least thousands of years. It has a long, even recorded history, and it's a much feared disease over the years, but it's the first infectious disease actually to have been eradicated, at least that we know of, by intentional human action. And so the irony of that is that the people who were developing the eradication campaign, people like D.A. Henderson and Bill Foege here in the U.S. at CDC, and Frank Fenner in Australia, and so on, had agreed that when smallpox was officially declared eradicated, they would destroy all the existing stocks, the various samples that were in a couple of places here in the U.S. and the Soviet Union as it was then, and so on. And then after smallpox was eradicated, it was D.A. Henderson who was very much one of the people in the forefront of the eradication effort, but also obviously a great advocate for destroying all those samples, sort of making, I guess, a great ceremony of our great success, a real triumph of public health, who pointed out that now that people aren't immunized routinely anymore, until 1972, everyone in the U.S. had to be immunized. Now, of course, after 72, because there was no natural disease threat, we stopped doing that. So there are a lot of susceptible people out there, and it was D.A. Henderson, oddly enough, who put himself in the odd position of saying, well, now that there are so many susceptible people out there and so few who are immunized, this has potential to be a real bioterrorist or biowarfare weapon. And unfortunately, there were people probably in defense establishments worldwide who had that sneaking suspicion that it might be possible. And so he found himself in the odd situation. Now nobody wants to destroy the virus because they're afraid that if they do that, they might not be able to discover new therapies or new diagnostic techniques. And so oddly enough, I really feel sorry for D.A. in the sense that he worked so hard with many other people to eradicate this, and you think it would be a great success we could all celebrate, but instead it's caused some other problems, which is the irony of eradication. It also means that you stop all the control measures you used to have, and you probably can't afford just to give it up.
Well, and the other issue is once you've eradicated something, if you stop vaccinating, then you stop developing the vaccine. So that's why we're now left with this vaccine that's what I would call vintage technology. I mean, this is really a very, very old vaccine that would never be approved today, and we're still using it because there just hasn't been any incentive for anybody to develop a better one because we haven't been giving it out much.
No, absolutely. The batches of vaccine that we had been using until very recently all dated back to 1982 or before. Same stuff I got when I was a kid. And because I used to go to Russia when I was working in the government, we had some collaborations with Russian scientists, including in the place in Siberia where one of the two declared stocks of smallpox samples is kept in Siberia. The other one, of course, is here at CDC. We had to be immunized, and I sort of got to remember what it was like. And it was also using that old 1982 vaccine, those batches, that date back a long way. But Acambis was the first attempt to make a new vaccine, with the concern that we might need to be able to produce vaccines again.
So this Acambis vaccine they basically took the New York State strain and I think plaque purified it and tested it and grew it in tissue culture, right?
Yes, essentially, there are a few strategies, and I think in the future they want to use a gentler, more heavily attenuated or more highly attenuated virus like the MVA because of concerns about possible adverse reactions and immunosuppression. But this is essentially just trying to reproduce the known technology, which was licensed for a long time. And it's ironic, we all got this. I'm old enough to have gotten this when I was a kid, and of course I don't even remember it. I have a scar to prove it, but I don't even remember it. And it may be that, luckily at that age, most of us survived. But of course, at that time, the threat of smallpox was such a major concern that we were willing to accept the occasional adverse reaction and other things. And now, of course, with the threat so indeterminate and probably low, we have a much lower threshold for adverse reactions if someone gets them.
Well, that's when you eradicate something, then the tolerance is low. It's the same with polio. It's eradicated in the U.S., so we lost our tolerance for vaccine-associated polio and went with an inactivated vaccine.
And I hope that won't be true of all immunizations. I know that there are an awful lot of very serious, used to be childhood diseases like diphtheria and pertussis or whooping cough, measles, which we tend to underplay. I had measles when I was a kid, but when I went to graduate school and learned about what measles actually does, we were told that's a very serious virus. And I guess I felt lucky, like most of my cohort, to have survived, but luckily we have a vaccine now, and these vaccines have saved many lives. So I hope we won't throw out the baby with the bathwater, as it were, and remember how important vaccines are in preventing really serious diseases that we may have forgotten about. Smallpox may be gone, but many of those others are still with us. You mentioned polio, for example.
Yep. Be interesting to see where that heads. We've talked about polio eradication here on TWiV before, and it's a difficult one. Okay, our next story is called Arsenic Exposure Suppresses the Initial Immune Response Against H1N1 in Mice. What do you think of this, Alan?
Interesting stuff that is just, I guess, just in press, if that's the term, about to go online at Science Today. We're recording this on May 22nd, so by the time this show goes live, this will be all out there. But I just got the backgrounder and paper today. It's still a draft edition of the paper. So this group, it's, let me just get the authors here. It's Joshua Hamilton at the Marine Biological Laboratory, Woods Hole. His lab did this. They had some preliminary data on arsenic exposure and immune responses in mice, and so they decided, well, let's look at do a real experiment with exposing the mice to various levels of arsenic in their drinking water, and then see how that changes their immune response against H1N1 influenza. And other than the scientific interest of whether this would work, there's also a very practical public health issue, which is that well water in many parts of the world, including many parts of Mexico, is commonly contaminated with arsenic. So this is not an industrial pollutant generally. It's a naturally occurring mineral in areas that have rock formations with a high arsenic level. In fact, up where I am in western Massachusetts, if you drill a well, there's a fairly good chance you'll get high arsenic, but we don't drink well water. So the issue is, if people in many parts of the world are exposed to high arsenic levels, is that going to affect their response to this virus? And the answer, at least if we're talking about mice, is yes, probably. So they exposed the mice to 100 parts per billion arsenic in their drinking water for 5 weeks, and then they gave them intranasal inoculations with sublethal doses of. It's a different H1N1. It's Puerto Rico strain.
It's the 1934 H1N1.
Yeah.
Yes.
A standard mouse-adapted strain.
Right.
Right, but still a similar enough virus. And they found that they had in these mice higher morbidity and higher virus titers in the lungs after infection and altered immune responses. So it's suggestive that arsenic, I know we already knew that arsenic is not good for you, but arsenic might be one factor in explaining what's been thought of as a high case fatality rate in Mexico for this virus. Now, of course, as we've discussed on the show, there are many other possible explanations for that, including the possibility that maybe there isn't really a higher case fatality rate in Mexico. But it was an intriguing explanation for that, and I thought a very interesting result.
It would be interesting. I don't know if anyone has gone back historically and looked at past outbreaks in places that had high arsenic levels. I know in parts of Taiwan they have very high arsenic levels, and actually high enough to have symptomatic disease, for example, having circulatory problems. As you know, one of the ironies is that in Bangladesh, there was a project to encourage people to drill these shallow wells called needle wells to improve the microbiological quality of the water so they wouldn't get sick from the water. But unfortunately, there's a lot of arsenic in the mineral underlying those wells. So they also get very high arsenic levels. Some of our colleagues here at the School of Public Health at Columbia, as a matter of fact, study that. And it would be interesting to know if there have been any clinical reports. I think if that's true, it's obviously not going to bode well for a lot of people in Bangladesh. I think I'll wait to see what actually pans out there. And I agree with you about Mexico. I think it still needs to be sorted out. But it's an interesting hypothesis, and it's interesting to see that you can have that sort of effect with fairly low levels of minerals. You may remember some years ago the work done on selenium deficiency and how it made mice and probably humans clinically in China, where there's selenium deficient soil, more susceptible to some viruses, particularly some of the small RNA viruses that could cause cardiomyopathy.
So do we know that there is arsenic in the water in Mexico?
In some parts of Mexico, yes.
It seems to me. So arsenic is an interesting effect on the innate immune system. It's actually been studied quite a bit, and we know it's bad for innate responses. So it would seem to me that it would be a problem for most virus infections to have arsenic in the drinking water. So it may be that our current susceptibility is already calculated by having arsenic in the drinking water. So I don't know how much this adds to it. I mean, as you said, Alan, it's not clear that what happened in Mexico is any more lethal than in any other part of the world. So right. I don't know how this factors into it. But, I think in the current climate, this gets a lot of attention, right? And I think they're using this mouse-adapted strain, what happens to be an H1N1, and they started this months ago before any of this happened. So they're at the right place at the right time.
Fortuitous timing.
Yeah. Yes.
Okay, so next we're going to talk about respirators. But before we do, let's have a word from our sponsors, Citrix, the makers of GoToMyPC. We've talked about this before, how it's always frustrating to sit in rush hour traffic waiting to get to the office so that you get to your computer and do your work. Well, if you had GoToMyPC, you wouldn't have to do that, at least not every day of the week. You could stay home and access your work computer from home. Do you commute from far, Steve?
I live on the Upper West Side, so it's about a half-hour commute by the traditional New York City method subway.
You have an easy commute.
I'm lucky there. It was harder when I had to go to Rockefeller University, which is way on the other side of town. It takes forever.
Well, if you ever want to work from home, you should consider GoToMyPC, which is a remote access program brought to you by Citrix, the award-winning service. You can instantly access all of your programs, files, email, even your network from home. It's all you need is an internet connection. You go to their website, a one-click install, no hardware, automatically, and then you can access your computer, in fact, from anywhere in the world. It's as secure as online banking. It's a great program. These people have done a great job at developing something that really helps. You can try GoToMyPC free for 30 days, but you must visit gotomypc.com/podcast. That's gotomypc.com/podcast for a free 30-day trial. Tell them TWiV sent you.
I have to tell you, a lot of the pandemic plans for influenza and other, severe emergencies suggest that people telecommute, that they stay home and
Sure.
work from home. And so much of it is on the computer. It sounds like this has a natural application.
That's a good idea. Good point. Yes, you don't want to be coming in if there's a pandemic, and this would be the key. I try and stay home, and I always fail because there's always something that brings me in. Respirators and masks do prevent flu transmission. Now, here on TWiV, we've been making fun of those flimsy masks. I think I have one in my drawer here. These flimsy masks, which really we think do no good. But now here's a study that says otherwise, Alan.
Yeah. So this just came up. Let's see. So the Lawrence Wine and Michael Atkinson at Stanford did this study. It's a computer modeling study, which is, of course, comes with all of the usual caveats, but they looked at the use of N95 respirators as well as surgical mask type of covers. And of course, the surgical masks are the ones that we always make fun of because it's really just kind of a glorified handkerchief that you strap over your face. The N95 respirators are tighter fitting, and they actually, in many cases, have an exhaust valve on them. You'll see construction workers who are sanding something that might be toxic will often be wearing these. They found in this article in risk analysis, the amount of protection that these could provide is actually significant. They modeled the spread of disease and found that you could actually potentially reduce your exposure by putting respirators and or masks on everybody. Now, of course, when the CDC comes out and says don't bother with respirators or masks, I think we discussed before, they're doing a somewhat more complicated calculation, which is that how many people are actually going to use these things properly. And I think the computer model in this case is assuming that most of these are being put on properly and kept on, which may be an overly generous assumption, but it's nonetheless an interesting finding.
So what do you think of this, Stephen?
It's interesting that back in 1918, during that very infamous pandemic, that's sort of our nightmare scenario, and hopefully we won't see again, but one never knows, that there was a very widespread use of surgical masks, and you can see pictures of policemen lined up with their surgical masks. Howard Markle at Michigan, who's a historian of this area, said that San Francisco actually mandated the use of surgical masks during the first wave of the 1918 pandemic, and people got so tired of wearing them that they used to have protests where they'd burn their masks. Finally, they lifted the restrictions when the wave passed, only to get hit by the more severe second wave. So, I think sometimes you have to be careful how you use these. There's been a lot of interest in masks and respirators, and CDC actually funded a few projects. We had one here. My colleague Elaine Larson was the principal investigator, and I was an investigator on the project, looking at whether masks and also hand hygiene would have an effect on household transmission of influenza. Ironically, during the time we had the grant that CDC was funding these grants, we had very light influenza season, so we never really got the answer. But we did find that in the absence of a really strong, something we talked about earlier, in the absence of a really strong sense of threat, people were very unwilling to wear masks simply because they're uncomfortable. You can't eat with them, of course, so you have to take them off. And we saw that with SARS. Hospital workers would wear their respirators, but sometimes they would make mistakes. And we have pictures that people took from Asia and other places of people wearing their N95 respirators and putting holes in them so they could smoke. And other things. Some of them make funny pictures, but obviously they're not good infection control. It's ironic that 90 years after, essentially more than 90 years after that great pandemic of 1918, we're still not sure what the best non-pharmaceutical control measures are. For example, it sounds very specialized, highly scientific, but if we assume that most of the transmission, as I believe, of influenza is probably by the respiratory route, Is it the large droplets or the fine aerosol particles? And that sounds like a very specialized argument, but surgical masks would be very good to protect you against the large particles, not so much against the small ones, which would travel further and where you'd probably need those N95 respirators. And that's still a question, which is a very interesting one. After all these years, we're still debating, how influenza actually infects people, and those have very practical implications, even though they seem like arcane scientific problems.
If you go to the CDC page, which is for the general public, they don't mention using face masks or respirators. Later on, if you go to the healthcare provider section, they will discuss it for people working in hospitals or offices. But for every person on the street, they don't recommend using them. And I think it's because, as Alan said, they don't often use them properly. So they have minimal effect and they give you some false sense of security.
Yeah, and people do find them uncomfortable. It's interesting.
Oh, sure.
We found in these studies that the CDC funded that, people said they wanted to wear masks or even, N95 respirators, but once they actually had to do it, of course, it might be different in a pandemic when people had more motivation, but it was very hard to actually get people to do it once they had to. They realized that it wasn't so comfortable after all.
There was a nice editorial or opinion in the New York Times a few weeks ago by a woman who, I think she was a physician, who said basically people, when you wear a mask, you can't see the person's mouth, and it's very important for communicating to see someone speaking, to actually see their lips moving, and it makes people very uncomfortable.
That's interesting. And of course, everyone points out how in places like Japan and other parts of Asia, people will wear masks. It turns out that very often they do it because they have allergies and they want to filter out the pollens. Right. But culturally, there's obviously quite a difference. It looks very different when someone gets on the subway here or on the street with a mask. Although I suppose it won't be so strange if we have a lot of flu cases. People will wear masks.
Do you think that isn't a mask more effective indoors where most of the transmission is occurring as opposed to walking on the street?
Probably. And, certainly for people who are in contact with a lot of other individuals, bus drivers, bank tellers. Interestingly enough, many of the companies that have made plans, financial companies being often multinational and having a lot of travelers among them, have actually stockpiled or at least put in their plan for more masks for people like bank tellers, figuring that they would have to change their masks from time to time during the day and refresh them, than for the executives who presumably have their own offices and never have to see anybody. But I think also one thing we learned during SARS was that people who have disease, very often a mask can be very effective if you put it on a sick person so they don't spread it to others. And they do that in a lot of emergency departments now when people who think they have the flu come in to be checked.
Our next story just came out yesterday in Morbidity Mortality Weekly Report, although it's dated May 22nd. That's today. Serum cross-reactive antibody response to the new influenza A H1N1 after vaccination with seasonal vaccine. So CDC has stored serum samples which were used to assess previous seasons' flu vaccine efficacy. They have it for 2005, 6, 7, and 8. They have pre- and post-immunization sera from children and adults. So they took those and did neutralization and hemagglutination inhibition tests to see if any of those sera reacted with one of the new strains. They used the California isolate, the new H1N1. And what they found is very interesting. First of all, none of the kids being from 9 months to 6 months to 9 years of age, none of them had any antibodies against the new viruses, either before or after immunization with any of those seasonal H1N1 strains. Some of the adults had some pre-existing antibody, and they were mostly in the older ones, and particularly the ones over 60 years old. And those adults also responded just twofold after immunization with the seasonal strains against this new virus. So the twofold increase is probably not protective, but they suggest that the pre-existing levels of antibodies in the neutralization test, the number is over 160. So that means at a dilution of 1 to 160, you're still getting 50% neutralization, I guess. So they don't know if that correlates with protection or even milder disease, but it could explain why a lot of the cases are in younger people. I think 60% of the cases are in people that are younger, according to CDC, and it could be that older people had an experience years ago with an H1N1 that's related to this strain.
I've been waiting all these years to get the payoff for having lived through the 1957 pandemic, which, of course, is what replaced the original H1N1, the one that were essentially the progeny of the 1918, domesticated and tamed over the years, but still varying to become the seasonal influenza. And that was, of course, wiped out by the 1957 pandemic. So I'm glad to know there was some payoff to that.
Yeah, you and I think both probably have some of that old H1N1. I being born in 1953, so I had a few years of exposure. Yeah. And so maybe we would have a milder infection. But this is interesting because for weeks we've been speculating about this. CDC has insisted that there is no protection, but Peter Palese a few weeks ago said it could make milder disease in older people.
Which would be very interesting because, as you know, when there's a heavy, not even a pandemic, but just a heavy seasonal influenza season, very often places like nursing homes and places where you have a lot of the elderly together just have tremendous problems with influenza. And the flu just goes right through the facility. And we haven't seen that, as far as I know.
Not at all.
With this particular
That's right. And it
strain.
may be, so this may partly explain it. Also, it could be that it just takes longer to get into the facilities, right? The older people don't socialize as much as the younger ones. But I suspect it has to do with previous exposure.
Well, it's nice to know that there is some benefit to experience. Being a gray beard,
to being old has some usefulness. Okay, our last story is something that Alan discovered just today, a Science Express article.
Yes, and this is breaking news. So this is just going out today, and looking at the draft now. So this one, it's sort of another example of how any research you're doing on H1N1 right now really ought to be pushed out the door at this point, because it's a very, very good time to be publishing on this virus. So this is just another analysis of the genomes. And as we've discussed, there are hundreds of these different genomes of the new H1N1 strains getting put into the databases, and it just dozens more every day, it seems. These researchers, Rebecca Garten and colleagues, went and probed through that database and tried to figure out what the origins are of this virus, which of course everybody would like to know for academic reasons as well as some public health reasons. And they found that this virus appears to have been circulating for a fair amount of time in swine, and the segments in it all seem to have originated in avian hosts and began circulating in pigs at various points from 1918 through 1998. And 6 of the 8 segments of the virus are from triple reassortant swine viruses. So these are viruses that have some material from human, avian, and swine viruses that reassorted. But of course, this is all happening in pigs, so they're still swine viruses. And it appears that this particular strain has been circulating in pigs in North America and Asia since about 1998. So when people are wondering, well, was it a particular factory farm that did this? In fact, this has been in the pigs in pretty much its current form for maybe more than a decade, at least based on this sequence analysis. The real question would be, what made this virus jump into humans and sustainably transmit in humans if it's been kicking around in pigs for all that time? And that, of course, is something that we don't really have a handle on at this point.
I think we should just clarify one thing, that this particular combination of segments has not been seen before. Having the 2 Eurasian segments, the M and the NA, hasn't been seen before. Each sequence, or each segment, if you will, has been in pigs for many years. And so that's the key observation here, is that these have been around and we didn't notice it.
Well, it's interesting. Influenza tends to do that, and we saw a very similar story with the notorious H5N1 or bird flu, which, as you know, caused a smaller outbreak, not easily transmissible person to person in Hong Kong about 10 years ago, and then sort of went underground, seemed to disappear. Well, all that time, of course, it was evolving in other species, mostly avian species, until it suddenly exploded again in 2003, both first in poultry and then in humans who were closely exposed. And that's another one that obviously has gotten a lot of attention, although it has not shown real human-to-human transmission to any sustained degree, luckily. And I think everyone was I think you should never be surprised by anything influenza does. It always defies our best attempts at prediction. But here we see an H1N1, which we had been paying a little attention to, but not a whole lot, suddenly becoming the next big influenza strain. But I think it shows that we've only recently started looking carefully in the animal hosts and animal reservoirs and doing surveillance there, and we really need to do a better job there. I know Rob Webster says he described a similar virus, triple reassortant, in 1998, which had been circulating around since the 70s.
Is that right?
Yeah. So this has been kicking around for a while, but obviously there have been a number of changes, and people often think, as you know, think of pigs as mixing vessels to allow influenza viruses from different species to mix and then perhaps more easily get into humans. I have a feeling that we will be paying a lot more attention to swine as well as the avian species.
And then some other species will turn out to be important as well that we didn't pay attention to.
Yeah, the shrews, ferrets.
Let me just read a couple of sentences from this manuscript. It's out now, by the way, so we can do this.
Good.
Blast searches on GenBank for each segment of the novel flu virus showed that viruses with genes of highest nucleotide sequence identity were isolated on average 10 years ago. So each of these the highest similarity were around already 10 years ago. These results indicate a lack of surveillance for many years in the original hosts of novel H1N1. So this is what we're saying. We should have been looking more in pigs, but it can't look everywhere, I suppose.
Well, I guess there's also a certain degree of, I hate to say it, complacency.
Of course. But we were focusing on avian strains. Yeah. There was a lot of surveillance. Here's another interesting sentence. Within each gene segment, there's a high sequence identity among the outbreak viruses sequenced to date, suggesting that the cross-species introduction into humans was a single event or multiple events of very similar viruses. So that's interesting because people have been speculating about whether it was one origin or multiple, but it's likely one or a few similar ones.
And as you say, what makes it interesting is why that particular event. Is it a particular variant, or was it just a particularly unlucky person or careless person? And as you know, there's been a lot of speculation for years, ever since 1918, about the origin of the notorious 1918 flu virus. Jeffrey Taubenberger has suggested that it was a direct introduction from avian reservoirs into humans, which normally would be extremely rare. We're quite resistant, unless it goes through another mammalian species like a pig first. But this may revive the thought that it could have, in fact, come through pigs.
Sure. This paper goes through a lot of the virulence signatures that have been looked at and which haven't been found in these viruses. And they also go over some antigenic studies. They say here, ferret antisera raised against this currently circulating seasonal viruses, that is last season's H1N1s, did not react with the novel strains. So further showing that these are antigenically quite different. And one of the explanations they set forth in this paper is that from 1918 up until about 1998, these H1s did not evolve extensively in pigs. They call it antigenic stasis. And it was only after that it began to evolve more. So there's a big gap from the human strains to the pig strains. So they say there's a lot of viruses in pigs which are very different from what we're infected with and which could come into us and cause problems because they don't cross-react. It's quite interesting.
Indeed.
So this is a nice paper which is just out. We'll put a link to in the show notes to this. And thank you, Alan, for bringing it to our attention. It's pretty cool that you get to have You get to see things ahead of time.
Yes.
Okay, I think that does it for our stories. And what I wanted to do for the remainder of our time is talk a little bit about what you do, Stephen, and how it relates to emerging infectious diseases and influenza. I suppose this is a good time to be talking about that because we have almost is this a pandemic or going to be, do you think?
Well, of course, the problem is that what we technically call a pandemic simply means a very widespread, worldwide, essentially geographic distribution of a virus or of an infection, obviously influenza in this case, which spreads person to person. But by technical definitions, yes, I think we could call it a pandemic. Over 40 countries have reported cases, and it's continuing to spread. Of course, the problem is that it's kind of a charged word for a lot of people who think of 1918 and other horrible events. So the WHO, for example, the World Health Organization, has been under some pressure not to call it a pandemic because it's bad public relations. So I think there the scientific terminology and the way we perceive it, the connotations the public perceives, are somewhat different.
You work on what we call pandemic preparedness. So what does that mean? And is every country prepared in the same way?
Well, no, I think countries differ in their degree of preparedness. And as you know, very famously, Indonesia complained in the case of H5N1 that they would be under-resourced for the next pandemic because where would they get the vaccines? Where would they get money for vaccine, which is, presumably ultimately what would stop the pandemic before everyone got infected. And I think that's a very real concern. These are worldwide events. That's why they're pandemics. But not every country has equal resources. In the United States, we have begun to get serious about it since about 2005. Ironically enough, then President Bush read John Barry's book, The Great Pandemic, about the 1918 influenza, and apparently was very impressed, at least by the portions he read.
You read a book?
Of the book, yeah.
I'm sorry, I was just expressing shock.
Yeah, well, I know. It's like that old joke about Claire Booth Luce and Pearl Mesta. Who wrote that? That's a great book. Who wrote it for you? And the other one said, I'm glad you liked it. Who read it to you? Well, in any case, however it actually happened, he did have the book in Crawford one summer in 2005, I think it was. And in that fall, actually announced that there would be a program directed towards pandemic preparedness. And I think although, obviously we can never do enough, it helped a lot in making people aware of it and making resources available. And we still have a long way to go, but I think we're better off now than we were in the past. Ironically, a lot of the things we're doing until the vaccine comes along will be basically what we did in 1918.
Is preparedness apply to a mild strain, as this one seems to be so far, or does it apply only to more serious, like perhaps an avian strain coming into humans?
I think most people obviously are concerned about more severe, more virulent disease. Obviously the nightmare scenario, as I mentioned earlier, is 1918, which we never hope to see its likes again. But nevertheless, that's the kind of thing you have to be prepared for, even if you think it's very low probability. It's obviously very devastating. Most of this particular pandemic, people have really experienced a sort of like a mild seasonal influenza, admittedly out of season, but that's another question. In most cases, I think there is a close parallel. These have been very mild disease cases, and I think the key right now is to do good surveillance and see if it becomes more virulent, comes back in a second wave, if it changes. And at that point, obviously, all the pandemic plans get kicked into much higher gear. But I think the fact that we've actually started to take influenza seriously and recognize that it has a lot of potential, even in a normal influenza year, thousands of people in this country die, it's not something to be taken for granted is, I think, an improvement over our previous attitudes. At least, we're not going to be fatalistic about it. Right now, I don't think we need draconian measures for this particular outbreak, but obviously need to watch and see what happens if it should change. One of the things I've personally been interested in fact, is the seasonality of influenza. And it turns out we think of it as in the temperate zones as a wintertime disease, basically. But in the subtropical areas, and of course people who live there have known this forever, all the time they've been living there. In places like Hong Kong, for example, we actually see two very good high peaks, one in the winter and one in the summer. In 2006 in Bangkok, the July peak actually far exceeded, in terms of number of cases, the winter peak, which of course in a tropical or subtropical area doesn't exactly correspond to our winter or summer. So I think it may have one pattern in temperate zones, but a lot of the things that we believe about seasonality, especially with influenza, I suspect are pretty glib and not yet really understood.
When we talk about surveillance in this context, usually it's what you were just discussing. It's following up and figuring out how the disease is spreading in as close to real time as we can. But to some extent, that's kind of reactive, isn't it? I mean, wouldn't it be better if we could predict when or where the next problem might come from? For example, we were just talking about these viruses that have been circulating in swine for a decade, and we didn't even know what their census was. Is that the kind of thing that could really be, I don't know, extended, or is the problem just too huge?
Well, I think ultimately it'll be, the problem is not insoluble, but it's a very tricky one because, all of our attempts to predict how influenza will behave have usually been unsuccessful, which is very interesting. So, the last time we saw a so-called swine flu was the famous example in 1976 when in Fort Dix, New Jersey, we had something over 200 cases of influenza, and then a young soldier who died. It turned out he had influenza, and he died actually because of overexertion, and the influenza probably contributed to that. But this led to a tremendous reaction to try to be proactive. There were immunization campaigns, and in fact, I remember getting my vaccine back then in 1976. And then when there wasn't a pandemic, of course, people focused on the downside, the adverse reactions that might occur from the vaccine. And it became a politically very embarrassing as well as very expensive problem. So I think we can learn a lot more and need to learn a lot more, but I think we're still some ways. There are a lot of us who are trying to work on developing better predictive models. But influenza is fascinating because, mundane as it may seem, it's still one of the in many ways least understood and hardest to predict, however much we may know about its genome. And it's, I think, very interesting that this is really the first time we've been able to get and have had the resources to get complete genomes from a lot of these viruses, so we can learn some interesting things about how the virus evolves as it goes through the human population, coming from animal sources and so on. But we still can't guess which of those animal viruses that we may see in pigs or in avian species are going to be the ones we should worry about. Until we can figure that out, I think it'll be useful to have the information because it may help us to figure that out. But we still don't know what to look for exactly.
One of our listeners sent an email asking about Africa. He said there haven't been any, or very few cases so far of H1N1, the new strain in Africa, and he was wondering if that's because there isn't sufficient surveillance, and then he wondered whether the pandemic preparedness there in that country would be suboptimal. Do you have any thoughts about that?
Well, I think it's a very good question, and I don't know why cases haven't been reported by and large from Africa. It's possible. Of course, different countries do give different priorities to surveillance, and you do find many more cases when you're looking hard for it. Many of the lower resourced countries are going to obviously need more resources if they have to deal with a serious pandemic. I think with all the serious diseases in Africa, what we've seen so far with H1N1 is going to seem pretty small by comparison, even if everyone gets it. But if it should get more severe, I think it'll be a whole different set of problems. South Africa and a couple of other countries are probably pretty well prepared, but there are many other countries in Africa, certainly, that are going to need a lot of help. And I think this is a very strong argument. We're all in the same world together, and we really need to work together to solve these problems.
So it sounds to me that you're not surprised that we have influenza through much of May now in the Northern Hemisphere.
No, and in fact, interestingly enough, many of our past pandemics, 1957, for example, started in July here in the United States. It may have started, in Asia. It probably started earlier. But it's interesting. We don't normally think of influenza as occurring during the summer, but we've seen pandemics breaking out late in the season. That seems usually to be when they happen, and I don't know why that is.
But seasonal influenza doesn't, right?
Yeah, it doesn't, and I think if it did in any significant numbers, all of us who ride the subways or go to summer school would be aware of it.
I read somewhere that you can isolate seasonal strains of influenza throughout the summer in big cities like New York.
I think that's probably true, and I know Peter Palese and others have suggested that the high humidity and higher temperature may make transmission less efficient, may require closer contact for efficient transmission, which is quite possible. I don't know. I think it's just one of those many things we still haven't learned, and obviously it's interesting in our sophisticated age that there are still a lot of really interesting questions that remain to be answered.
I think it's not unlikely that you have low-grade influenza throughout the summer. It's not noticed. It's a cold. People think it's a head cold or something, and that's how the virus is maintained over the summer in some ways.
Yeah, it wouldn't surprise me either. Certainly this H1N1, if it hadn't been for the severe whatever caused that outbreak in Mexico, which led them to take very strong measures, closing schools, closing public places. We might not even have noticed this H1N1. There was a school, St. Francis, which was the first one in New York City to have recognized cases. And I think in the end there were about 148 lab-confirmed cases. It's thought there were about 200 actual cases that probably would be lab confirmed if you tested them all. It's obviously a burden to do that many samples. But when the New York City Health Department did a survey, they found that 1/3 of the students at that school actually had recently had an influenza-like illness. We don't know what it was, but it could have been the H1N1 too.
This brings up an interesting question. Earlier this week, CDC published their latest graph on isolates from the U.S., and they show a peak of isolations of influenza virus in February and then decline in April to very low levels, and they're isolating H1N1s and H3N2s and influenza B viruses. And then suddenly, of course, in April, week 17 and 18, a spike in the number of viruses, not just the new H1N1s, but all the old influenza strains are re-isolated again, so the peak goes right up again. So what is that because of increased surveillance, or is it really that all the strains are coming back for some reason?
No, I don't actually know the answer. As you know, many laboratories and many health departments sort of do their surveillance in this part of the world during the traditional flu season, so they consider it perhaps wasteful of resources to be doing a lot of surveillance, especially since it's not that easy. Most cases of flu probably don't get to the attention of health departments or physicians because we take them for granted and they're just so familiar. But I think a lot of places just simply don't test outside the normal flu season. So even if it existed, it would take a special effort, like CDC going out and doing a lot of testing.
Let's move on to our last two parts of TWiV. And one of them is email. We get lots of listener email. So let's read a couple of these. The first one is from Len, who said, I wanted to pass along the following link. It provides an interesting article from Nature News on growing seed strains for influenza vaccines. Your other listeners might find it interesting. And he gave a link to an article which explains how the influenza vaccines are made scientist by the name of Doris Bucher at New York College of Medicine, who you may have heard of.
Yes.
Is involved. She makes the high-yielding recombinants. She takes the isolate provided by WHO, which she says she got in April, and she makes a high-yielding recombinant with the PR8 strain. And so it grows well in eggs, but has the surface antigens from the current strain. So she's working on that now. And I remember when I was a student at Mount Sinai, a PhD student, she was there. And Ed Kilbourne, the chairman of the department, used to do that. Now, apparently she's inherited his recombination making
Yes, I
machinery.
think he passed the mantle on to her. And Ed Kilbourne, I think, was in fact the person who discovered or invented that technology.
Right.
And it certainly was a big breakthrough in the 1950s when it was first developed in being able to develop a vaccine against influenza. The irony, of course, is that we have more rapid, larger scale biotechnology today, which has not been applied to influenza. There have only recently been attempts to develop tissue culture vaccines. There is an attenuated live virus vaccine, which is more expensive, but I think has a lot of interesting promise. But that too has to be grown in eggs by the traditional method, and that's terrific, except it's really hard to get enough eggs of the right quality that would pass FDA. So these are not just ordinary fertilized eggs. They come from very special chickens, and you just can't get them to make the eggs any faster. So that's really a limitation. And it would be nice to be able to use some of our newer biotechnology, whether it's tissue culture or even the cloned hemagglutinin protein, which after all is what we're purifying out of this material and using as our antigen. So it's kind of a long roundabout way to do it. But at least I'm very glad we have something that does work.
Well, as you know, those alternatives are ongoing in research labs, so we'll find out. Interestingly, Ed Kilbourne was responsible for getting me to go to Mount Sinai, where I eventually worked with Peter Bellesi, but that's a story for another time.
Ed Kilbourne's an old friend, and I think he deserves a lot of respect for the fine work he's done over the years in influenza.
Absolutely. It's too bad he's not listening. So Len had a question. Can you address the concept of amplification as it relates to pandemic influenza viruses? This is the concept that a virus that passes rapidly through a number of hosts of the same species can build virulence as it does so. John Barry in The Great Influenza suggests this as a possible partial explanation for such nasty virulence for the fall 1918 outbreak of Spanish flu.
Yeah, this was originally suggested by Paul Ewald, who thought that there's a correlation if you more rapidly cycle an infection through the human population and it can spread more readily, it could become more virulent. And that's sort of just taking the well-known relationship that's been worked out with some pox viruses and others, namely that if a virus kills its host too quickly before it can be transmitted, it obviously isn't going to go anywhere. So it can afford to be as virulent as its transmissibility allows, because it has to get to a new host before it kills its original host. And so Paul Ewald took that idea, sort of turned it on its head and said, well, it works in the other direction too. If you passage something quickly and it's highly transmissible, then it could gain in virulence, which is a very interesting idea. I'm not quite sure. It's very hard to prove it. There are a few examples, and I don't know in the case of 1918. We've had a lot of speculation. And it's just hard to say, because obviously we did have it cycling rapidly through the human population and soldiers and very close contact. So it's hard to say. It's kind of if I could use a trite cliche after our last discussion, a sort of a chicken or egg problem. But it's certainly an interesting theory, and I think bears thinking about in this context.
My feeling is viruses evolved to be able to replicate well and be transmitted.
Yeah.
And whatever it takes to achieve that, they will do. So it may be more virulent, so you cough harder or sneeze harder, or it may not be. I don't think you can necessarily hypothesize that it has to increase in virulence.
Well, I agree with you, and I agree with you. The viruses that are successful, by definition, are the ones that evolve to grow well and transmit well from host to host. And of course, some of the most transmissible, like the common cold, caused by various rhino and enteroviruses and others, are among the mildest infections, even though they may be highly transmissible.
Sure. Well, there are examples where viruses evolved to be less virulent. So you remember probably in Australia the pox virus that was released to try and control rabbits. After the first year, evolved to a less virulent form to coexist with the rabbits. The rabbits also evolved as well.
Well, and I think as Paul Ewald also said, or suggested, the route of transmission makes a big difference as well.
Sure.
If you have a transmission route that can work well with an incapacitated host, then that certainly would be conducive to greater virulence than one that requires the host to be out talking to other hosts.
Right.
Still a lot of interesting questions to discover. The Myxoma study, the study with the rabbits in Australia, is quite fascinating too. It reminds me of something Lewis Thomas wrote in one of his essays, and I'm not going to get this quite exact, so I'll paraphrase it. He says something to the effect that serious, highly virulent, highly lethal disease with a microbe is an example of an incomplete negotiation between the microbe and the host.
That's right. Yes, I remember that too. Yeah, I have the book here on my shelf. Incomplete negotiations, or something like that. Yeah. I'll look it up and put it in the show notes.
Yeah.
All right.
Or unfinished negotiation, something like that.
All right, let's read one more email and then wrap things up here. Professor Ricardo writes, first of all, thank you for this wonderful conversation you guys allow us to hear. I'm a university teacher in Portugal, and the combination of new information as well as an historical background about virus helps a lot during lecture. All of you are quite good at storytelling, which in my perspective is a very important tool in our business. Well, thank you very much. And Ricardo suggested 2 iPhone apps that we might want to look into. One is called CDC Alert, which apparently gives the CDC disease alerts.
Wow.
So you might want to look at that, Stephen. And the other is PhD Comics.
Yes. I don't read it on my iPhone, but an absolute classic.
Yeah, that's an old one. It's been around for years. Filed higher and deeper.
I have read some of my friends sometimes send me some episodes of PhD Comics. I think there was one that shows a chart of salaries, and it proves that the head of the athletic department or the best coach in the college usually makes far more than any professor and even the president.
No doubt about that. That's true.
But I'll have to look into the CDC alerts. That's great.
And now his question. I've been listening, more devouring your podcast, and there is always the vaccination question. In discussion with some colleagues, and my position is totally in favor of vaccination, I don't have arguments to rebate the problem of overexposure to vaccines, taking too much vaccines like rotavirus or varicella. As far as I know, we are bombarded every day with thousands of antigens. 1 or 2 more wouldn't make a difference. But then the question of the place where the vaccine components are introduced, blood, muscle, rises the possible nasty immune reactions that can come from vaccination. Any help here? My best regards and keep it coming. You are very good. So he's worried about, putting antigens in non-natural places causing odd reactions. Any thoughts about that?
It's an interesting question. And, ironically, as we introduce some newer vaccines, there are a lot of vaccines we used to give that we don't give anymore because we don't think they're necessary. But, we discover, for example, in Eastern Europe, when the Soviet Union fell apart and there was no more money for a lot of the public health efforts, they stopped immunizing against the sort of classic childhood diseases, the ones I mentioned earlier, diphtheria and pertussis and so on. And they saw a tremendous increase, which, of course, got them back to immunizing. Well, I think, vaccinology is more an art than a science. Let's face it. It's an attempt to stimulate the immune system. The fact that it's been so successful is remarkable, but I think I have much the same feelings that your listener does. I'm in favor of immunization. I do recognize there's some degree of artificiality to it. I think that a lot of the harm that people may attribute to immunizations may be coincidental, but clearly there are rare adverse reactions. And it would be much better to be able to do what we're doing more now, have either highly purified or highly attenuated live virus or live vaccines, which we're doing now with chickenpox, with measles for many years, mumps, rubella, of course, the MMR vaccine, with typhoid even. And it certainly is a lot better to get that vaccine than to get typhoid. Sure. I can attest to that. But maybe that's the wave of the future. I don't know.
I think you're right. I think that, for example, flu mist, which is the flu vaccine that you spray, is more like a natural infection. And we'll probably move towards those. Polio vaccine, of course, has always been a natural route of immunization.
Yeah, and part of it, I think, is that we don't really understand what, in the jargon of the vaccine field, they call the correlates of protection. In other words, which immune responses are really the important ones to try to encourage for any given infection. And I'm sure they must vary with different types of infections. If we understood the immune system better in that respect, we might be able to do a better job.
It's time for our science picks of the week. Now here, Stephen, what we do is we try and pick something scientific to recommend to our readers, a book, a blog, a website, a museum, a TV show. So Alan and I will have some, and maybe while we're talking, you can think of something you like very much that you want to share with our listeners. Alan, why don't you go first?
Okay. In keeping with my technical and somewhat odd choice from last week, my pick of the week for scientists and for anybody who's interested in organizing large amounts of information is something called org mode, which is it's actually a mode that runs within another program called Emacs. And Emacs is possibly one of the oldest pieces of software that's still in common use. This is a text editor. It's available for all platforms. And so Emacs is a text editor. You can write any kind of text in it. It's used a lot to write computer programs. I commonly write my articles in it. You can take notes in it. It's a very, very, very powerful way to edit text. One of the great things about Emacs or any other text editor is that unlike a word processor, it saves things as plain text. So 50 years from now, if somebody wants to open an old file that you created, this is about as close as you can get to an archival file format. I don't know if you've had the situation where you go back to files you created 10 years ago with an old version of Works or Word or something, and you can't open the files anymore. So org mode runs on top of that, and it allows you to do project organization. If I were in the lab today, I mean, you remember I was one of the first in the lab to keep sort of an electronic notebook, and I did it in Microsoft Word because that was what was available. Looking back, I should have used Emacs, and if I were doing this today, I would do it with org mode. It allows you to do things like you can create a to-do item, and to create it, you just type an asterisk and then in all capitals the word to-do. And then you type in what you need to do. So as you take notes, you can actually put in items that need to be done, and then the program can collate all of this, and you can even export it into things like iCal. So you can get very nice project management going on. And it's all saved as plain text, which means that you will be able to go back to these in the future, and or future generations of scientists will be able to read your notes.
I've heard of this. I think there is another version called text.org. Something like that. Basically the same idea. It's a text file for getting things done, or day planner, notebooks, organization. I think Gina Trapani of Lifehacker is working on something related to this as well. So this is quite a popular approach to this sort of organizational stuff.
There is a major learning curve to it, but
yes, I've heard.
Yes. Once you get past that, it really, really is worth it.
Well, we have some listeners who probably love this sort of thing, any of you who have come over from the TWiV network. But the rest of us can also learn as well. So that's pretty cool. Great. Orgmode.org.
Orgmode.org.
Thank you.
And I have that pasted into the agenda.
Very good. All right. My choice is a website on swine influenza genetic analysis. It's a rather long URL, so I would mention it here. We'll put it in the show notes, but it's basically a site where they take the sequences that are coming out on a daily basis, and they make phylogenetic trees, and they do analysis. And from the page itself, These pages exist to enable the rapid dissemination of results resulting from the analysis of genetic data from the ongoing Influenza A H1N1 outbreak attributed to swine flu. So basically, you can see really nice trees, reassortment history, molecular epidemiology, molecular clock analysis, etc. So it's a bit technical, but interesting nonetheless. Have you got a science pick or something that you would like to share with our listeners, Stephen?
Well, it's hard to resist. This is, as you know, the 200th birthday of Charles Darwin.
Mm-hmm.
And, I think we all owe a great debt to Darwin. He still remains a controversial figure, except among most scientists who, I think, accept evolution as a given. And The Origin of Species, I think, holds up very well and has a lot of very interesting ideas that are timely even today. There was a book, I haven't read it yet, but one that I just saw in the magazine called The Scientist, which talks about 10 great innovations of evolution, actual innovations in life, which promises to be very interesting because it talks about some of the complex functions that we normally think, people ask, how can evolution do this? And but somehow it manages. And he talks about such things as vision and consciousness, which you'd think would be beyond the scope of science. Lewis Thomas's essays, I find, still hold up very well. And as you know, the Rockefeller University here in New York put up a prize called the Lewis Thomas Prize for the scientist as poet, for good science writing, and by scientists, which I'm sorry to say is rare enough probably because we're not encouraged to get too literary when we publish. But the first recipient was Lewis Thomas, and they're going to give this year's award to Robert Sapolsky, who's a neurobiologist and works on, among other things, baboons, and is a wonderful writer. A little off the subject of virology, but I think very, very well worth reading.
That's fine. It's all kinds of science at this point, right? Not just virology. Well, thanks very much. That's great. I was just thinking the other day that I need to reread The Origin of Species.
Josh Lederberg, in that 1989 Emerging Viruses Conference, pointed out that humans, to a large extent, are a man-made species in the sense that, we create culture, we create a lot of artifacts to insulate us against some of the, what would otherwise be selective factors in the outside world.
But that's part of evolution, right?
Absolutely. That's
no matter. I mean, I always view whatever we do is part of our evolution. We're applying selection in its own way.
Yeah, and I don't think we can stop evolution in its tracks. It may go into stasis in some cases, in relative equilibrium with some viruses and natural hosts, but eventually, we see evolution continuing.
And for people who are interested in reading Origin of Species, of course, you can do it for free. It's long past copyright. It's available from the Gutenberg Project. If you don't mind reading it on a computer screen, I really don't recommend trying to read a book that long on a computer screen, though. But if you have an e-reader, I just saw on Amazon.com the Kindle version of the book is available, and it's free as well. So you can just download it automatically into your Amazon Kindle if you do that.
Thank you, Alan.
That's a bargain.
Stephen, thank you so much for joining us.
Thank you. It was a pleasure to be here
and
to chat with you.
I hope you will return someday.
I'll look forward to it.
Perhaps one day when Dick is here, we have you together and you can really have at it because the two of you together are amazing.
You're very kind. Thank you. It would be fun. I'd look forward to it.
We will put a link to your website in our show notes so people can know where to find you and read more about you.
Thank you.
Alan, thanks again.
Always a pleasure.
Alan on Twitter is Alan Dove. Alan writes a blog at
sporadic blog
at dovedocs.org.
.com.
Sorry. You can find me on Twitter at PROFVRR. Actually, it's the TWiV Twitter. And I also write a blog called virology.ws, which I spend far too much time on because viruses are captivating, and the opportunity to explain them to people is captivating as well. Don't forget, you can find more science podcasts at sciencepodcasters.org and promednetwork.com. Send us your questions and comments. Tell us what you want us to talk about. TWiV at twiv.tv. You can also send us an audio file, an MP3 by email, and we'll play it on the show. We will be back next week You've been listening to This Week in Virology, the podcast about all kinds of viruses. Thanks for joining us. See you next time. Another TWiV is viral.